Yes, everyone. So sorry for the delay, bachcho. But now I am live, so I hope that I am clearly audible and clearly visible to you. Yes or no? Hi, Shivesh. How are you? So sorry, Shivesh. I am little late. But let's quickly start the class now. Tell me that am I audible or not? Okay. Am I audible? Am I visible? Yes. For me, I am audible and visible, and I think so for you all also, guys. I am audible and visible. Okay. So today in this session, what we have decided to start with some of the lessons, which was the genetic material. Search for the genetic material. You remembered any doubts in the previous session, guys? Any doubts anywhere in the previous session? Let me know in the comment section. Mark your attendance first of all, everyone. Mark your attendance first of all, everyone. I can see Shivesh and the other students. Where are they? कहाँ हो सारे लोग? Okay, chalo. So guys, today what we know is that genetic material is what? We know that for today the genetic material is DNA. Yes or no? But before that, there was lots and lots of confusion between the genetic material. Whether the genetic material was RNA, whether the genetic material was DNA, whether the genetic material was protein. So there were lots and lots of confusion regarding this. So series of experiments happened, and with the help of these series of experiment, what we have learned that DNA is the genetic material. How these experiment happened, which scientists did this experiment, what questions can come from these kinds of experiment, we are going to check it today. Okay. Everyone ready for the class? Everyone ready for the class? Great. Chalo. Great. So let's start with the genetic material. So what should be the property of genetic material? एक genetic material कैसा होना चाहिए? How should it be? First of all, let's talk about this. So number one thing, that it should be what? It should be stable. Yes or no? It should be stable. And stable means what? Both physically and both structurally and chemically. Stable both structurally and chemically. Okay. Both structurally and chemically it should be stable. Yes or no? Second thing is that what? Second thing is that should be able to express protein banana chahiye. Yes or no? Should be able to express themselves. Like how, sir? See, if there is a genetic material, that genetic material contains some information. Let's suppose there is a genetic material, and this genetic material contains the information of eye color. So this genetic material must be what? Must be able to give this information so that this eye color should be represented. Yes or no? If it is not able to express itself, then why we will be calling it? What's the use of the genetic material? Yes or no? Yes? Any doubts? Any doubts anywhere? Great, Shivesh. Great. So the genetic material should be able to express itself. It should what? It should be what? It should be stable both structurally and chemically. Great. Now let's move on and let's talk about the third point, which our genetic material should have. They should able to what? They should able to replicate themselves. अपना copy बनाने आना चाहिए इनको. Should be able to replicate itself. Okay. Should be able to replicate itself. Fourth point should be what? Fourth point, they should undergo slow mutation. slow mutation should be present. Why, sir, slow mutation should be, is mutation harmful or necessary? So see here, in some cases mutation is very necessary. Maybe with the help of mutation they are able to generate, they are able to have such a character which is going to help in the near future. So slow mutation is needed. Fast mutation is not needed. Fast mutation will be what? The characters will be changing in lots and lots of less duration, so that will create what? That will create problem. Slow mutation, good mutation should be happening. The genetic material should not be very resistant that nahi mutations तो करवाएंगे ही नहीं, that should not be there. Mutations होना चाहिए but बहुत ज़्यादा mutations नहीं होना, ऐसा नहीं होना चाहिए कि कुछ बहुत बड़ा changes हो गया तुरंत पे, right? Changes आए but small and slow changes, okay, which could be helpful, right? चलो, great. Now it should be stable. It should be stable enough. ऐसा नहीं कि बच्चे में, अगर हम छोटे बच्चे हैं तो the genetic material is something else, बड़े हो गए तो फिर change हो गया, और बड़े हो गए तो फिर change हो गया. So it should not change with the age. Yes or no? Should not change with the age. Should not change with the, should not change with the age. Okay, clear to everyone? Any doubts? कोई doubts यहाँ पे? If any doubts, let me know in the comment section, people. Any doubts, let me know in the comment section. No doubts? Great. चलो, now let's see here. If I talk about the RNA, and DNA, which one should we consider as a better genetic material? RNA और DNA में से कौन सा genetic material better होना चाहिए? See, RNA is what? RNA is not very stable. Yes or no? It's not very stable. Less stable, I should say. RNA is what? RNA is the less stable. But if I talk about the DNA, so DNA is what? DNA is more stable. DNA is what? DNA is more stable. And if it is more stable, guys, and if it is more stable, this should have what? This should have the chances of, this should have what? This should be the genetic material. Yes or no? Yes, sir. Now, if I talk about the RNA, RNA was the first genetic material. If someone asks you, in this whole world, which was the first, which molecule was behaving as the genetic material for the very first time? So for the very first time, guys, RNA was the genetic material. RNA was the very first genetic material. So let's write here that RNA was what? RNA is the first genetic material. RNA is the first genetic material. But guys, if I talk about DNA, so DNA is what? DNA is the, like DNA, the genetic material as DNA came after the RNA, but it is more stable. Yes or no? Clear to everyone? Clear? Okay. Great. Now, if I talk about some of the cases like bacteriophage, like plant virus, TMV plant virus. So in these cases, the genetic material is what? The genetic material is RNA only. RNA as a genetic material is present in some organisms. RNA as a genetic material, is present in some organism. In what organism? Let's write the example. So first of all, TMV में बच्चों, TMV virus, tobacco mosaic virus में जो genetic material है वो क्या है? RNA. And if I talk about the bacteriophage, so in bacteriophage also, guys, what type of genetic material do we have? The genetic material is RNA. Yes or no? The genetic material is RNA. Clear to everyone? Yes or no? Okay, great. So these things we have studied about the RNA and DNA, like why RNA is not a perfect genetic material, why DNA is the perfect genetic material, because DNA in DNA what is happening? Deoxyribose sugar is present, which is more stable than the RNA. Yes or no? So that's why we consider that DNA is the more stable genetic material, while RNA is not the most stable genetic material. But where there is the absence of DNA, guys, where there is no DNA, then who will be the genetic material? Then the genetic material will be the RNA. Clear to everyone? Any doubts anywhere? Any doubts anywhere? Great. Chalo. Good. Now, let's talk about the search for genetic material. Let us now talk about the search for genetic material experiment which proved that what is the genetic material, whether it is the RNA or whether it's the DNA. So search for genetic material. Let us see that what was the experiments which helped to prove that the RNA is not the genetic material, the DNA is the genetic material. Yes or no? Okay. So first of all, experiment by Griffith. Transformational experiment by Griffith. We have what? We have an experiment which is transformation experiment by Griffith. Okay, by Griffith. What is this transformation experiment by Griffith? See, what happened? He took what? He took 2 strains of Streptococcus pneumoniae. He took 2 strains of Streptococcus pneumoniae. Okay, what is the strains? Strains is basically the variety. Okay, 2 varieties. Now we are going to say it as strains. 2 varieties. Now we are going to say it as strains of what? Of Streptococcus pneumoniae. It's a bacteria. Streptococcus pneumoniae. Okay. Yes or no? Now, this bacteria is what? This bacteria is responsible to form what? This bacteria is responsible to produce a disease which is what? Which is pneumonia. Okay. Now what happens here? 2 strains were there. What strain? 2 strains. One is the smooth S strain and the other one was the R strain. 2 strains were there. One the S and the other was the R strain. Okay. So see here what did this Griffith did. So first of all, he took this S strain bacteria. Okay. S strain bacteria. S strain bacteria was also known as smooth bacteria. Now what smooth, sir? See, if I talk about the smooth meaning here, so the covering of the bacteria was what? It was smooth. It was covered with the polysaccharides. Yes or no? This type of bacteria was known as S strain or the smooth strain bacteria. Right? Okay. Now this smooth strain bacteria was injected to mice as it is. ये bacteria लिया गया और इस bacteria को क्या किया गया? इस बैक्टीरिया को कहाँ डाल दिया गया? इस बैक्टीरिया को डाल दिया गया माइस में. So this smooth S strain bacteria was injected where? Was injected to mice. And what happened, guys? Mice died. Yes or no? Mice died. Here the mice died. One second, I'll write it. Just give me a second, guys. What happened? Am I stuck? No. So mice died. Okay. Now, second thing what happened is that, he took what? Second case, he took R strain bacteria. He took which kind of bacteria, guys? He took R strain bacteria. Yes or no? Okay. Now, when he took R strain bacteria, फिर से इसने क्या किया? फिर से R strain bacteria को he injected it into the mice. He injected the R strain bacteria into healthy mice. Okay, the mice was very healthy. He was not having any type of problem. He doesn't have to give NEET, so it was very, very healthy. And to Griffith's surprise, this mice survived. So what conclusion we can make here, guys? What kind of conclusion we can make here? Tell me. यहाँ से हम क्या conclusion बना सकते हैं? What kind of conclusion we can have? The conclusion which we can have here is that S strain is what? S strain is something which is causing death, which is virulent, which is causing the disease. Yes or no? Because when we injected S strain bacteria into the mice, what happened? The mice died. But when we injected R strain into the bacteria, sorry, into the mice survived. That means nothing happened to the mice. Yes or no? So S strain bacteria is virulent. ये बदमाश बच्चा है. S strain क्या है? ये बदमाश है. It is virulent. It is disease causing. Yes or no? It is virulent. It is disease causing. And S strain is what? S strain is non-virulent. It is not causing disease. It is non-virulent. Yes or no? Any doubts still here? Any doubts still here? Shivesh, any doubts still here? Great. No doubts. Great. Now let's move on. Let's move on and let's talk about the things further. Now what happened? Griffith was like, now I'll do some real experiments. So Griffith did what? He took S strain bacteria. He took S strain bacteria. Hi, review on point. How are you? It took S strain bacteria. Now if I talk about the S strain bacteria, guys, what he did is that he heat killed this bacteria. He heat killed this bacteria. Okay, heat killed this bacteria. Heat killed bacteria. Okay, so S strain को लिया, S strain को क्या करा? पानी में डाल के उबाल दिया, मर गया ये S strain. Yes or no? S strain is heat killed. Now what happened? Now what happened? It again injected to the mice. They again injected it. That's great review on point. Again injected in mice. Okay, again injected in mice. Now what happened, guys? Mice will die or survive? Mice will die or survive? Let me know in the comment section. According to you, the mice will die or survive? What we have done? We have just taken the S strain bacteria. We have not, actually Griffith has taken. So Griffith has taken the S strain bacteria, and that S strain bacteria is now somehow heat killed. And when that heat killed bacteria is injected in the mice, what is going to happen? Shivesh is saying that mice will die. Mice का RIP हो जाएगा. Shivesh is saying that mice का RIP हो जाएगा. So guys, mice will survive here. Mice will survive here. Okay, mice survived. Mice survived. Now what happened? He did another experiment. What experiment? See, 4 cases. He did 4 cases. 4 cases he have done. So 3 cases we have seen. Let's talk about the 4th cases. So in 4th case, what he has done? He has taken S strain. He has taken S strain. Okay, and this time this S strain is what? It is heat killed. It is heat killed. Yes or no? It is heat killed. But guys, with the in presence of this heat killed, the Griffith has also taken one more strain, which is R strain. Now you remember that this R strain was that bacteria which is not causing the disease. R strain was that kind of bacteria which was not causing the disease. Yes or no? Yes. But this time what happened? When these were inserted, when these were injected to the mice, what happened? Mice died. Mice का क्या हो गया? RIP हो गया. Mice died. So that means what we are having here we saw that earlier R strain was not causing any disease, but when mixed with S strain, R strain somehow became virulent and mice died. Yes or no? Yes or no? So Griffith said that what? Griffith concluded that something from the Griffith conclusion. Let's write here Griffith conclusion. What was Griffith conclusion? See here. So Griffith concluded that something was present in the S strain which got transferred to the R strain. Great Sri Sai. Something is present in the S strain which got transferred into the R strain, and what happened? And it changed the characteristic of R strain, and R strain got virulent. Yes or no? So see here, Griffith experiment, too much work. Okay. No worries, Sri Sai. See here. So what happened here is that Griffith said that something from S strain, got transferred, got transferred, got transferred where? To R strain, to R strain, and which lead what? Which lead the conversion of R strain to S strain type, and finally mice died and caused the death of the mice. Yes or no? And caused the death of the mice. Okay, so what we saw here is that R strain और S strain जब अलग अलग लिए गए थे, when R strain and S strain was taken individually, then what happened? Then during the S strain the mice died, but R strain mice survived. Now what? Now S strain heat killed. When S strain was heat killed, mice survived. But when S strain heat killed S strain injected with the living R strain, mice died. So that means for the conclusion, if you would be there and I would be there, then also we would have got this conclusion that due to the presence of S strain, R strain got the properties of virulent and killed the mice. Yes or no? Because ये तो मर गया था, S strain तो मर गया था. That means there is something which is going from S strain to R strain and causing the death Yes or no? Yes or no? Clear to everyone? Any doubts anywhere? Let me know in the comment section. फटाफट से. If you have any doubts, let me know in the comment section. No doubts? No doubts? Great, Srishai, great. Sorry, Shivesh. Okay, chalo. Let's move on. But Griffith was not able to. Griffith was not able to. Sorry. Griffith was not able to conclude that whether it was DNA, whether it was RNA, whether it was protein. Yes or no? Yes or no? Okay, great. Chal. Now let's talk about the another experiment which was done by Avery, McLeod and McCarthy experiment. Okay, another experiment, experiment 2. Missing Srishai. Yeah, Shivesh and Srishai, like the name is quite similar, na? So that's why sometimes I mix both of your names. Chalo. So experiment 2. Let's talk about the experiment 2 here. What experiment 2? Number 1, the name is Avery, McLeod and McCarthy experiment. Okay, Avery, McLeod and McCarthy experiment. Avery, McLeod and McCarthy experiment. What happened here? They repeated the Griffith experiment and confirmed that transforming material was DNA. Okay, they confirmed that transforming material was DNA. कैसे करा देखो, कैसे करा, how he has done it, how they have done it. See here how they have done it. One second, just a second. So they used the three enzyme. One enzyme was RNase, second enzyme was DNase, and the third enzyme was protease. Okay, protease. Yes or no? 3 enzymes they have taken. Now what they have done, see here. They took what? They took this S strain. They took this S strain plus R strain. Okay, S strain plus R strain. Yes or no? S strain plus R strain. Is there any doubt, guys? Is there any doubt to anyone? सब ठीक चल रहा ना? Am I audible clearly? Tell me. All good na? Okay. Just let me know in the comment section, guys, if it's all good. बताओ मुझे. Yes, all good. Okay, great. Then let's move forward. See here. So what happened here is that what happened during the this one, which is the every McLeod and McCarthy experiment. So what happened? We took R strain, okay? We took R strain and SS strain. Yes or no? We took R strain and SS strain. And we took what? We took these different enzymes. RNAs लिया एक बार DNAs लिया एक बार protease लिया. तो transformation कब हुआ? Transformation RNAs के केस में भी हुआ. RNAs is what? It just cut the RNA, digest the RNA. What is this RNAs? What's the use of this RNAs? Digest the RNA enzyme. What is the use of DNAs? Digest the DNA. What is the use of this protease? Digest the proteins. Okay. So transformation didn't take place when DNAs was used. When DNAs was used, transformation nahi hua. That means R strain didn't got converted into S strain. R strain didn't got converted into S strain when the enzyme DNase was used. When the enzyme DNase was used. So this proved that something which is DNA is something which is what? Which is responsible for the transformation. Yes or no? Which is responsible for the transformation. Clear to everyone? Clear to everyone? Any doubts? Any doubts, bachcho? No doubts? Great. So this was done by the Avery, McLeod and McCarthy experiment. Yes or no? So they proved that DNA is the kind of genetic material, but still what happened now? There were many biologists who didn't believe this. So there is what? There is again another set of scientists who did some experiment and proved that, guys, DNA is the genetic material. So let's check the final proof. Let's check the final proof whether the DNA is genetic material or not. Okay. So see here. The another experiment was done by the Hershey and Chase. Whenever I say Hershey, now I remember this thing. What thing, sir? I remember this Hershey syrup. Okay, chalo. Let's move on. Is my video lagging, guys? नहीं बच्चों, सब तो ठीक चल रहा. Everything is going fine. चलो. Hershey and Chase experiment, exactly. Hershey and Chase experiment. Now what they did? They did the final thing and they said that कि नहीं भैया, तुम लोगों ने जो DNA वाला देखा है वो सही बात है. That is very true. DNA ही क्या है? हमारा genetic material है. So what happened here? See, they took radioactive. सबसे पहले तो they took what? They took bacteriophage. Okay. They took what? They took bacteriophage. Yes or no? Yes, Srishai. Bacteriophage. And if I talk about the bacteriophage, so what they did is that they labeled this bacteriophage with what? With radioactive phosphorus. Okay. They used what? They used the what I was saying. They used the radioactive phosphorus. Okay. Radioactive phosphorus. ठीक है? They used what? They used the radioactive phosphorus. Clear? Okay. Now, in one culture, they also used what? They also used the sulfur also. Okay. So they used radioactive. One second, what happened? They used radioactive phosphorus and they used radioactive sulfur. But कब कहाँ कैसे use किया? Let's check that. Yeah. Why radioactive, guys? कुछ और क्यों नहीं use करा? Radioactive क्यों use करा? Tell me. Radioactive क्यों use करा? See. Because it's easy to trace, na? Radioactive इसलिए use करा because it is easy to trace. Yes or no? Because it is easy to trace. Now, let's check about this. 1 second guys, something is happening or what? चलो चलो चलो, let's see. So we have this bacteriophage, कुछ ऐसा दिखता है bacteriophage. Let me try to make this bacteriophage for you. ये हमारा प्यारा प्यारा सा bacteriophage, okay? Yes or no? Light करना था? Okay, दिवाली के लिए. Okay, yeah. Nice idea, Shrishai. Nice idea, right? Everything is fine? Okay, great. Great, chalo. So, Shrishai is having a new best friend, Shivesh. But, unfortunately, it's online, right? Chalo. Now, I'm happy that we are building a family here. You guys won't forget me, na? NEET nikal jayega, फिर then we'll have, who was Harsh sir? चलो इसके थोड़े पैर वैर भी बना देते हैं. So this is what I have taken two cases. I have taken two cases. One is what? In this I have labeled the sulfur as the radioactive. I have labeled sulfur as the radioactive. Okay. And in this case what I have done? In this case I have labeled phosphorus as the radioactive. Okay. Now we know that guys, sulfur is what? Sulfur is present in the protein coat. Yes or no? Sulfur is present where? Sulfur is present in the protein coat. And if I talk about the phosphorus, so phosphorus where it is present? It is present in the DNA. Yes or no? Yes. Okay. Now we did 3 steps. We did what? We did 3 steps. First one is infection. Even from college you're gonna join my class. Okay, great. Then I'll be like meet my ex-student guys. Okay, chalo. So when first of all we will do the infection. Now this is bacteriophage. What is it? It is bacteriophage. Yes or no? They are bacteriophage. Okay. So bacteriophage do what? Bacteriophage करता है क्या भाई? ये E. coli में infection कराएगा. Yes or no? Bacteriophage क्या करता है? E. coli में infection करेगा. So see, we took this E. coli, and we have allowed to do the infection. Okay. So ये bacteriophage क्या करता है? ऐसे आके बैठता है यहाँ पे like this and सारा का सारा genetic material इसके अंदर डाल देता है. Okay. Yes or no? After this, after the infection is done, what we have done? Blending. हमने blend कर दिया इसको. What we have done is blending. Yes or no? Yes or no? चलो great. तो blending करने के बाद I'll change the slide. Shivesh is saying I hope this channel grows. Thank you Shivesh, thanks a lot. It means a lot. Then I'll also flaunt. Oh my god, I have millions of subscribers. Yes or no? Chalo. Then we will be having blending. Blending में क्या करा? Like we did the blending so that कि properly the bacteriophage and all can be attached. Yes or no? Blending में bacteriophage and all can be attached. We have done blending. And third we have done centrifugation. Yes or no? So in centrifugation, when we did a centrifugation, what happened? In our test tube, what we saw two things. एक चीज तो नीचे बैठा हुआ था, which is called as what? Which is called as 1 second. Retentate. Okay. Something is happening. Something is lagging, guys. Is everything fine? 1 second. I'll open my phone. 1 second, guys. Yeah, it's totally fine. Now it's lagging. Yeah. Okay. Something is happening. Something is lagging, guys. ये लैग वैग मेरी क्लास में क्यों होता है? उमा शंकर सर के क्लास में तो बिल्कुल नहीं होता ये लैग. बताओ. 1 second बच्चों. 1 second. अरे क्या हो रहा है ये? It's fine. चलो great. चलो ग्रेट. ओके सो देन वी डिड व्हाट, वी डिड सेंट्रीफ्यूगेशन. तो ऐसा समझो ना कि एक टेस्ट ट्यूब लिया था हमने, जस्ट अंडरस्टैंड इट लाइक दिस दैट वी हैव टेकन अ टेस्ट ट्यूब. ओके एंड in this test tube what we have done, in this test tube we have allowed the bacteriophage to cause the infection. Okay, सब चीज इसी टेस्ट ट्यूब के अंदर हुआ. now blending करने के बाद what we have done centrifugation. and when Uma Shankar sir also had a lag today. Then I am satisfied. Then I am satisfied. Chalo, I'll ask Uma sir how was the lag experience, because it happens in my class generally, right? Chalo. So, do you want, guys, that we and Uma Shankar plan something good for you all? Like, do you want, should we have Q&A session on Sunday? Something like that. Do you want that? Should we do it? Chalo, see here. So, centrifugation happened and हमें क्या मिला that there was no radioactive detected in the retentate part. नीचे में कोई भी radioactivity नहीं था. No radioactivity detected here. But in above part where the protein coat was present, here radioactivity were present. Supernatant में क्या था? Radioactive present था. Supernatant में what happened? Radioactive was present. So in supernatant protein was there, protein coat was there, yes or no? Protein coat was there, and here what? Cell was there, DNA was there, yes or no? So in this case, it is sure that protein is made up of sulfur and protein is not the genetic material, yes or no? Yes or no? Now, in the second case, what we saw? In the second case here, what happened that when radioactive phosphorus is used, when radioactive phosphorus is used, then guys what? Then it was shown, it was observed that the DNA part was radioactive. Then it was radioactive, and in this case the supernatant was not radioactive. In this case the supernatant was not radioactive, yes or no? Clear? So इससे क्या प्रूव हुआ? Yes, obviously. So इससे क्या प्रूव हुआ? What happened? क्या प्रूव हुआ इससे? बताओ. इससे क्या हुआ बताओ मुझे. Clear? Okay, that's great. Okay, Shivesh is saying we will meet you one day. Okay, good Shivesh, great. Good. I recently traveled to Delhi and I met some of my students. It feels very good for me also, like teaching. DNA was the genetic material, exactly. Sitting on the screen, behind the screen and teaching and meeting those students in a real life, it really gives good experience. Okay, chalo. Which was transferred, exactly. Yes. So clear to everyone. So search for genetic material is clear. Any doubts? I'll just summarize this thing. So first of all, we had what? We had Griffith, yes or no? First of all, सबसे पहले कौन था बताओ? Griffith uncle. ये पेन का thickness इतना मोटा कैसे हो गया? First of all, it was Griffith, right? चलो. First was Griffith. Okay, Griffith did what? क्या किया था Griffith ने? बताओ जल्दी. What this Griffith did? R strain and S strain experiment. Yes or no? R strain and S strain experiment. R strain and S strain experiment. Yes or no? Yes, sir. Then what? Then came what? Then came Avery McLeod and McCarthy experiment. Yes or no? What they did? Yeah. इन्होंने क्या किया था? 3 एंजाइम यूज किया था, आरएनएज, Yes, exactly. RNAs, DNAs and what? And protease. Yes or no? और ट्रांसफॉर्मेशन कब नहीं हुआ था? जब प्रोटीएज, क्या स्पेलिंग लिखते हो सर आप? 1 सेकेंड. प्रोटीएज. ओके, ट्रांसफॉर्मेशन कब नहीं हुआ था? ट्रांसफॉर्मेशन कब नहीं हुआ था बताओ? ट्रांसफॉर्मेशन कब नहीं हुआ था? जब डीएनए डाला गया था. यस, जब डीएनए डाला गया था तो ट्रांसफॉर्मेशन नहीं हुआ था. यस और नो? यस. जस्ट इमेजिन शिवेश एंड श्रीशा, यू विल गेट द सेम कॉलेज देन. हाँ, वही मिल लेना कॉलेज में. यस और नो? सो प्रिपेयर वेल, क्रैक एम्स न्यू दिल्ली. यस और नो? यस. ओके, चलो. सो ट्रांसफॉर्मेशन कब नहीं हुआ? ट्रांसफॉर्मेशन तब नहीं हुआ जब डीएनए को यूज करा गया. क्लियर? अब उसके बाद फिर कौन आया? उसके बाद फिर आया, आई थिंक सो, अगेन आई एम लैगिंग. टंग 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 टंग. चलो, उसके बाद कौन आया? उसके बाद हमारे पास जो एक्सपेरिमेंट करने आए वो कौन थे? यस, लेट्स गो टू एम्स. लेट्स गो टू एम्स. मैं क्या करूंगा जाके? हाँ, तुम लोग जाओगे ना? चलो. यस. Now what? Now there was one more experiment which was done by Hershey's and Chase experiment. Yes, and I'll write here only. Hershey's and Chase experiment. Hershey and Chase experiment. इन्होंने क्या किया था? रेडियो एक्टिव फॉस्फोरस एंड सल्फर यूज किया. रेडियो एक्टिव फॉस्फोरस एंड सल्फर यूज किया. यस और नो? रेडियो एक्टिव फॉस्फोरस एंड सल्फर. क्लियर? इस चीज की स्क्रीनशॉट ले लो. जितने भी एक्सपेरिमेंट्स उनका हमने एक पेज में लिख लिया. आई नो माई हैंडराइटिंग इज पैथेटिक, but you guys will manage, right? इसकी screenshot ले लो and it will help you to revise. Yes or no? No one from Manipur has gone to AIMS. Shrishai, I will help you a lot and let's break this thing. Okay? Let's break this thing. Let's think that you should be the very first person to go to AIMS from Manipur. It will be a proud moment, such a proud moment it will be, right? Yes or no? Yes or no? Clear? So मेहनत करते हम लोग, पहुंचेंगे हम लोग एम्स पे. ठीक है? हाँ, ओके, चलो, ग्रेट. स्क्रीनशॉट ले लिया इसका? गुड गुड. सर गिव योर इंस्टा. Okay. It's because of the physics and chemistry teacher of Manipur. We have a wonderful physics teacher in our channel, which is Uma Shankar Sir. Yes or no? He is very experienced and he is studying in the IIT. So his level of teaching you can understand. And we are also launching the, बता दूं क्या? मेरी टीम ने मुझे नहीं बोला बताने को. फिलहाल मेरी टीम ने मुझे नहीं बोला बता. चलो मैं बता देता हूँ. Very soon we will be launching a chemistry teacher also. Very soon. And he is also very good. He is also very good. So biology, physics and chemistry all done. And then we will meet in AIMS New Delhi. Yes or no? Yes? Yes or no? Clear? Great. ओके, चलो चलो. नो वरीज गाइज, नो वरीज. पढ़ेंगे, हो जाएगा, सब कुछ हो जाएगा. मुझे बताओ यहां तक कोई डाउट? एनी डाउट्स चलिए गाइज? एनी डाउट्स चलिए? कोई भी डाउट है? ओके, ग्रेट. नो डाउट्स. चलो. सो लेट्स इंडियन ट्रेन टू स्कोर 700. ओके ओके, ग्रेट ग्रेट. चलो. नाउ लेट्स टॉक अबाउट द कंक्लूजन. इन सारे एक्सपेरिमेंट से कंक्लूजन क्या निकला? शिवेश इज स्टॉकिंग मी. यस और नो शिवेश? सारे चीजों में, इन सारे चीजों से कंक्लूजन क्या निकला? व्हाट वाज द कंक्लूजन गाइस? नो डाउट्स? ओके, चलो. ओनली See what happened na, Hershey's and Chase experiment mein, in Hershey's and Chase experiment we know that only DNA can enter, yes or no? Only DNA can enter, पूरा का पूरा बैक्टीरिया तो enter नहीं हो जाता, only DNA can enter. So only bacteriophage DNA can enter the bacteria, yes or no? Yes. So when phosphorus as radioactive, or I can write when radioactive phosphorus is taken, then what happened? Then the retentate was what? Then the retentate was radioactive. Then the retentate was radioactive. Yes or no? Was radioactive. So this was the conclusion made from the Hershey's and Chase experiment, and in this way we figured it out. Yes, the DNA is the genetic material. Clear? Okay, chalo, great. So tomorrow onwards guys, we are going to have all the basic classes of this chapter is finished. सारे के सारे basic चीजें इस chapter के हमने खत्म कर लिए हैं. कल से we will be starting what? We will be starting the main chapter. So don't miss those sessions, okay? Don't miss those sessions. Clear? Okay, Shivesh? Insta चलाना बंद करो शिवेश. अब इधर आओ. Okay, chalo. So till then, bye bye everyone. Let's meet tomorrow, and tomorrow we will be starting what? We will be starting the central dogma, and finally hum translation, transcription and production of DNA, all these things we are going to study. सारे processes हम कल से start कर देंगे. Okay everyone? चलो. Till then, bye bye everyone. See you in the next class. And we have to crack AIMS Delhi this time. Yes or no? Yes, Shivesh and Shrisha, you guys have to crack AIMS New Delhi. Chalo. Bye bye everyone. See you.